Hello students, nice to meet you in our final revision. Please concentrate with me. Let's start. First question. Choose the correct answer for each of the following. The sequences of nucleotides in a messenger RNA is necessary to determine the space. Is it amino acids in protein, codons in DNA, nucleotides in the gene, nucleotides in the anticodon of transfer RNA? Again, I'm going to say this question again and please concentrate with me. Sequences of nucleotides in messenger RNA is necessary to determine amino acid in protein, codons in DNA, nucleotide in the gene, nucleotide in anticodon of transfer RNA. Of course, the answer is going to be the sequence of nucleotide in a messenger RNA is necessary to determine the amino acid in protein. Next, parthenogenesis occurs in all of the living organisms except space. Crustaceans, worms, insects, or sponge. Of course, the answer is going to be parthenogenesis occurs in all of the following organisms except sponge. Testosterone hormone is secreted from space. Cooper's glands, seminal vesicle, Sertoli cells, interstitial cells in testes. Good. Testosterone hormone is secreted from interstitial cells in testes. The theory of Huxley states that when the skeletal muscle contracts, the advance sets of space are withdrawn by help of energy. Is it transverse links, myosin filaments, actin filaments, myosin and actin filaments? Again, the theory of Huxley states that when the skeletal muscle contracts, the adjacent sets of space are withdrawn by helping of energy. Is it transverse links, myosin filament, actin filament, myosin and actin filament? Of course, the answer is going to be actin filament. Diabetes mellitus can be caused by space, insufficient insulin, lack of insulin receptors, obesity, all the above. You are right. Diabetes mellitus can be caused by all the above. The two centrioles existing in neck of human male gamete play a role in the division of fertilized ovum within in the space: ovary, fallopian tube, uterus, or vagina. The two centrioles existing in neck of human male gamete play a role in the division of fertilized ovum within in the space. Is it ovary, fallopian tube, uterus, or vagina? Of course, the answer is going to be the two centrioles existing in neck of human male gamete play a role in the division of fertilized ovum within fallopian tube. Which of the following represents a recognition sequence of restriction endonucleases? Is it 5' GGCC3', and on the other side it's CCGG5', or 5' AGTC 3', on the other side 3' TCAG, or 5' A, C, C, A, 3' on the other side, 3' T, G, G, T, 5'. The last one, 5' A, A, G, G, 3' on the other side, 3' T, T, C, C, 5'. Of course, the answer is going to be, it's number A, which is 5' G, G, C, C, 3', 3' on the other side, C, C, G, G, 5'. Let's move to another item of questions. Mention uses of each of the following. DNA hybridization. Good. It's used to detect the presence of a particular gene and its genome, and also determine evolutionary relationships between different species. Mention the uses of recombinant DNA in agriculture. Recombinant DNA in agriculture used to produce crop plants genes for resistance to herbicides, also isolate and transplant the genes that enable members of legumes house nitrogen-fixing bacteria in their roots, then transplant the relevant genes into other crop plants and set them up with bacteria. Would eliminate the need for nitrogenous fertilizers. And also mention the use of colchicine. Colchicine is used to induce a desired mutation by causing atrophy, a death of the growth or growing tip cells in plants, and the new tissue are regenerated underneath the dead cells. These new tissues may contain some polyploidy cells. I'm going to say that again, and please concentrate with me. Mention the use of colchicine. It's used to induce desired mutation by causing atrophy, death of the growing tip cells in plants, and the new tissues are regenerated underneath the dead cells. These new tissues may contain some polyploidy cells. Another item of question, write the scientific term, the organ through which the urethra passes. Yes, penis, yes. A method of sexual reproduction in the primitive organisms in which the contents of the cell fuses with contents of another cell. You are right, it's called conjugation. The hormone antagonizes the action of parathyroid gland hormone. Yes, you are right, it's called calcitonin. A plant portion that if it is don't find what is its text during its movement, it wilts and dies. Of course, this portion is called tendril. The distance between each successive two Z lines in the skeletal muscle. Good. The distance between each successive two Z lines in the skeletal muscle is called sarcomere. What are differences between budding in yeast and sponge? Let's see. Budding in sponge and budding in yeast. First, budding in sponge. The bud grows as a cellular protrusion from one side of the body due to division of interstitial cells and their differentiation to a bud. This bud grows gradually to resemble the mother entirely. It usually separates to start its independent life. But budding in yeast, the bud arises as a lateral projection from the original cell, while the nucleus divides mitotically. into two nuclei. One of them remains in the other cell while the other moves towards the bud. It grows gradually and may remains connected with the mother cell till its full growth. Then it separates or continues in connection. The mother cell forming cellular colonies with it. I am going to say this comparison again and please concentrate with me. Budding in sponge. The bud grows as a cellular protrusion from one side of a body due to division of interstitial cells and their differentiation to a bud. This bud grows gradually to resemble the mother entirely. It usually separates to start its independent life. But budding in yeast, the bud arises as a lateral projection from the original cell, while the nucleus divides mitotically into two nuclei. One of them remains in the other cell, while the other moves towards the bud. It grows gradually and may remains connected with the mother cell till its full growth. Then it separates or continues in connection. The mother cell forming cellular colonies with it. So now compare between the structure of cerebral part and facial part of human skulls. Let's see. Skull's facial part includes face bones, the two jaws, and the two positions, the positions of sense organs, ears, eyes, and nose. But skull's cerebral part consists of 8 serrated bones attached firmly to each other. Its posterior contains a foramen magnum, through which the spinal cord connects to the brain. I am going to say this comparison again. Compare between the structure of cerebral part and facial part of human skull. Skull's facial part includes face bones, the two jaws, and positions of sense organs, ears, eyes, and nose. Skull's cerebral part consists of 8 serrated bones attached firmly to each other. Its posterior contains a foramen magnum, through which the spinal cord connects to the brain. Explain each of the following. Unlimited numbers of proteins are formed in bodies of organisms, although number of amino acids does not exceed 20. Difference of proteins is due to difference in number, kinds, and arrangement of amino acids in polymers, attributed to number of polymers that form the protein, and also weak hydrogen bonds that may give the protein molecule its special shape. I'm going to say this answer again, and please concentrate. Unlimited numbers of proteins are formed in bodies of organisms, although number of amino acid does not exceed 20. The answer is going to be difference of proteins is due to differences in the numbers, kinds, and arrangement of amino acids in polymers, attributed to the number of polymers that form the protein, and also weak hydrogen bonds that may give the protein molecule its special shape. Next, when cloning DNA sequences, it is better to use cells in which the gene required to deal is active as pancreatic cells. Again, explain. When cloning DNA sequence, it's better to use cells in which gene required to deal is active as pancreatic cells. Yes, in these cells there is great deal of messenger RNA carrying the message necessary make the proteins, where this nucleic acid is isolated and used as a template to make complementary DNA by using the reverse transcriptase enzyme. This enzyme produced a single strand of DNA. Its complement can be synthesized by DNA polymerase. I am going to say this answer again, and please concentrate with me. In these cells, there is great deal of messenger RNA carrying the message necessary make the proteins where this nucleic acid is isolated and used as a template to make complementary DNA by using the reverse transcriptase enzyme. This enzyme produced a single strand of DNA. Its complement can be synthesized by DNA polymerase. Give reason for stop of menstrual cycle during pregnancy. Yes, because during pregnancy, the corpus luteum or placenta secretes progesterone hormone which inhibits ovulation. I'm going to say the answer again. Because during pregnancy, corpus luteum or placenta secretes progesterone hormone which inhibits ovulation. Next, obviousness, the phenomena of alternation of generation in the life cycle of Plasmodium. Because life cycle of Plasmodium has a sexual generation that reproduce by gamete in mosquito, followed by a sexual generation that reproduce by sporogony and schizogony in man. Sporogony in mosquito and schizogony in man. I am going to say this answer again and please concentrate. Because life cycle of Plasmodium has a sexual generation that reproduce by gametes in mosquito, followed by asexual reproduction generation that reproduce by sporogony in mosquito and by schizogony in man. Next, the ability to adapt with environment is reduced in the individual that reproduce asexually. Because the individual resulted from asexual reproduction resembles the origin individual from which they were resulted, so the features of the following generations remain the same, even if surrounding conditions change at change in the environment. Most offspring become exposed to destruction unless their parents had been adapted for that change. I'm going to say this answer again and please concentrate. The ability to adapt with environment is reduced in individuals that reproduce asexually. Because the individual resulted from asexual reproduction resembles the origin individual from which they were resulted, so the feature of the following generations remain the same, even if the surrounding conditions change at change in the environment. Most of offspring become exposed to destruction unless their parents had been adapted for that change. Next, cartilage usually found at the tips of bones, especially at joints. Cartilage usually found at the tips of bones, especially at joints. Because cartilage protects the bones from corrosion due to continuous friction. Next, when somebody is infected with measles, he will not be infected again. Yes, due to acquired immunity against this disease, because after first infection, body forms the memory B and T cells, can live for tens of years and may survive till death. These memory cells store information about these antigens. So the same individual infected by the same disease again, the memory cells form in the past, from the past, respond to the pathogen once it enters the body, and they start divide quickly to produce within short period of time large amounts of antibody active T cells for killing microbe, so symptoms does not appear. I'm going to say this answer again. Due to acquired immunity against this disease, because after first infection, body forms the memory B and T cells that can live for tens of years and may survive till death. These memory cells store information about these antigens. So same individual infected by the same disease again, memory cells from in the past respond to the pathogen once it enters the body, and they start divide quickly to produce within short period of time large amounts of the antibodies active T cells for killing the microbe, so symptoms does not appear. Another item of question. There are different types of ligase. Some of them have a role for DNA replication and some have a role for DNA repairing. Explain these statements. Again, and please concentrate. There are different types of ligase. Some of them have role for DNA replication and some have role in DNA repairing. Explain this statement. Yes, during DNA repairing, working in harmony can recognize and remove a damaged area of DNA and replace it with nucleotide complementary to those on the other strand which is damaged. I'm going to say this answer again. During DNA repairing, it's working in harmony can recognize and remove a damaged area of DNA, replacing it with nucleotide complementary to those on the strand opposite the damaged portion. Let's move to another item of question. Rewrite the following statements after correcting the underlined parts. The gene responsible for formation of hemoglobin is situated in chromosome number 9. Of course, the answer is going to be the gene responsible for formation of hemoglobin is situated on chromosome number 11. Ligase is used in PCR machine for clone DNA piece. The answer is going to be tag polymerase enzyme is used in PCR machine for clone DNA piece. Next, nucleosomes are heterogeneous group of proteins. Yes, non-histones are heterogeneous group of proteins. In eukaryotic cells, ribosomes are produced inside the cytoplasm. In eukaryotic cells, ribosomes are produced inside nucleolus. B cells formed and complete their growth to become mature in the thymus gland. Of course, T cells formed and complete their growth to become mature in thymus gland. Not B cells, it's T cells. What is the importance of endosperm tissue? Yes, supplies the early developing embryo with food. Mention the site and function for each of the following. First, sepals. Of course, it's outer wheel of flower. Their function, they protect the inner parts of the flower against drought, rain, or wind. Mention site and function of vagina. Site of vagina starts from the cervix to the genital opening. Its function secretes mucus fluid to moisten. It has folds to allows its expansion during birth. Mention site and function of Sertoli cells. Site of Sertoli cell inside each seminiferous tubule. And function, they secrete a fluid to nourish the sperms inside the testes. It is supposed that they give also immunization function. Another item of question. What happens when separation of a piece from chromosomes during division and rotates 180 degrees around itself and rejoins the same chromosome again? Yes, a chromosomal mutation occurs. What are the difference between the components of nucleosome and the nucleotide? Yes, component of nucleosome, DNA is wounded around clusters of histone, forming a string of particles. but components of nucleotide, a 5-carbon sugar, a phosphate group, and a nitrogenous base. What can you expect in each of the following cases? Absence of cholinesterase enzyme from the neuromuscular junction. Acetylcholine is continued, so membrane permeability to ions does not return to the resting state, and it becomes not ready to be stimulated. Another respond again. Next, absence of glenoid cavity from the pectoral girdle. The humerus bone cannot be attached to the scapula bone, and shoulder joint doesn't form, leading to difficulty in the movement of upper arm. What is meant by each of the following? Sarcomere. Sarcomere is a distance between each successive 2Z lines. The motor unit. The motor unit is the functional unit of the skeletal muscle because the muscle contraction is the summation of all motor units forming the muscle. Each motor unit consists of a group of muscles fibers and the nerve fibers supplying them. Explain the sex of newborn can be controlled in farm animals. Yes, the sex of newborn can be controlled in farm animal in which sperms with chromosomes are separated from the sperms. Chromosomes with X chromosomes are separated from the sperms with Y chromosomes by laboratory means such as centrifugation or exposure to a limited electric field and using or one type of sperms to produce females or males. Again, I am going to say this answer again. The sex of newborn can be controlled in farm animals in which the sperms with X chromosomes are separated from the sperms of Y chromosomes by laboratory means such as centrifugation or exposure to a limited electric field and using one or type of sperms to produce males or females, males only or females. My dear students, I wish you all success, good luck, and goodbye.